Good afternoon, everyone. Thank you so much for joining us for the last session of the Pathologists Overseas and ASCP Laboratory QMS course. This is our 20th session, and it's being presented by Dr. Emily Glenn on documents and records. And Emily is one of our Pathologists Overseas members and organizers. She's responsible for a lot of the AP Pathologists Overseas work. So it's really appropriate that she's finishing us up. As always, remember that if you have questions during the presentation or during the scenario, please put those in the Q&A box, which is at the bottom right of your screen. If you have a technical problem, you can put that in the chat. But also, if Emily, while speaking, asks a question of the audience that she wants feedback from you to get your thoughts or your feedback, you can also put that in the chat, not in the Q&A. And that way Ken can follow along and give her some feedback as she's talking to you. Timothy Amokili, who is also with Pathologists Overseas, has also joined. He's disguised as another Emily Glenn, but he is here in the background and will be helping with the Q&A and the discussion part. This is being recorded, and as always, it will be live on the loading page within 24 hours. The session from Tuesday is there now. Someone had emailed about it, but it is live now, so you should be able to see it and you shouldn't need a passcode. We will, after the conclusion of this will be the last session, but in about 2 weeks, about the second week of June, we will be sending out certificates of participation, and we also will have some follow-up surveys. One will be an evaluation of the course, and the other one will be an evaluation of just your knowledge, for us to see how you did and how you enjoyed the course. So with that, I'm going to turn it over to Emily, and go ahead.
Great, thank you so much, Dan, and thanks everyone for participating in our series. So as Dan mentioned, I'm one of the co-directors at Pathologists Overseas, and I'm also a hematopathologist at the University of Washington, and I'll be talking to you today about documents and records. So after the lecture, hopefully the goal is that you'll be able to explain the difference between documents and records, describe the hierarchy of documents and the role of each level, outline the contents of a quality manual, outline the content that should be included in a standard operating procedure, explain the elements of a document management system, and describe methods and tools to properly store documents and records. So these are the 12 components of the quality management system, and here is documents and records. So why do laboratories need to manage documents and records? Really, it's because we need to be able to find the information whenever it is needed. So just like at a library, there's a way to organize all of the books. Laboratories really need to find an organizational framework for documents and records so that when they need to find that information, they can easily access it. So documents are information about policies, processes, and procedures. They need to be easily accessed by anyone who needs it. Information must be updated and maintained. So these are really living documents. If things change, then documents need to be changed to reflect that. They establish standardized forms. So, creating templates that will be used for recording or reporting information within the laboratory. And examples of documents include quality manuals, standard operating procedures, and job aids. Records, in contrast, are information that are produced by the laboratory when performing or reporting a laboratory test. They also need to be easily accessed or retrieved. And this information is permanent. So this is really a documentation of what happened in the laboratory. So these do not change. Standardized forms or documents in the form of standardized forms become records when they're utilized to record information. And examples of records include those completed forms, charts, sample logs, QC information, and patient reports. So first we'll give an overview of documents. So as I mentioned earlier, documents are policies, processes, and procedures. So policies really refer to what to do. So according to CLSI, policies are a written statement of overall intentions and directions defined by those in the organization and endorsed by management. So they really organize what the lab does in sort of a general way and provides kind of a foundation of the lab's mission, their philosophy, their goals, and purpose. They provide a framework for the quality system and should always be specified in the quality manual. So an example of a policy is the quality manual. Processes are how it happens. So processes are a set of interrelated or interacting activities that transform inputs and outputs. So they basically take the framework that was created in the policy and describe the steps that are involved to carry that out. An easy way to represent processes are flowcharts, and they involve essentially a series of steps that occur over time. So an example of a process would be a flowchart. So this is what they mean by inputs and outputs. So essentially a process describes how an input or a specimen, so basically the point of obtaining the specimen when a patient presents to the laboratory to have their blood drawn, what activities occur in the laboratory to generate a laboratory result from that sample, and then the output is the result. And so the process is basically the flow of the specimen through the laboratory, and there could be several procedures within this process. So procedures are essentially how we do it. So there are specific activities of a process, and the typical procedure is known in the laboratory is known as an SOP, or a standard operating procedure. And these are step-by-step instructions for performing a single activity. Abbreviated forms of SOPs are known as job aids or work instructions, and these can be placed at the bench for quick reference, and they are meant to supplement but do not replace an SOP. So in this process, you can have several SOPs, right? You have an SOP for how to draw the blood, an SOP for how to process the specimen, an SOP for actually how to run the CBC, and then another one for how the laboratory results are going to be communicated to the LIS. So each of these processes within the laboratory can have multiple procedures embedded within them. So this just highlights what I just explained. So this is known as the document hierarchy. So policies, or what we do, are sort of the root of the laboratory. They really form the foundation. They set the tone for the quality system that the laboratory is operating under. Processes represent the trunks. So that's really how it happens. So it's sort of how you get from the processes to the procedures. And then the procedures represent the leaves. So this is really the nuts and bolts of how processes are carried out. So why are documents important? So in the most simplistic sense, written documents are required by formal laboratory standards. So accreditation bodies, regulatory bodies, they're going to ask to see copies of your written documents. But on a broader level, they're really the essential guidelines for the laboratory. They're the way that we communicate. So for example, a quality manual provides the overall guiding document for the quality system within the laboratory. SOPs provide the step-by-step written instructions for how the tests are actually performed. Reference materials are the sources of information, both scientific and clinical, as well as methodological, for how these SOPs and quality that inform the SOPs and quality manual. So as I said, documents are really the way that we communicate within the laboratory. And for that reason, they really must be written down. So I think we've all been in situations or heard of situations where someone approaches you, one of your coworkers approaches you and says, "Oh yeah, why are you doing the test that way? Remember we had that lab meeting and the manager was there and we decided we're not going to do the test that way anymore. We're going to make this change." But you weren't aware of that change. And so this creates, when things aren't written down, they create a situation where some of the staff don't know that there was a change because they weren't at that meeting. So they're doing the test the old way. Or, even the folks who were at the meeting, they may be heard incorrectly or they forgot some steps. And so they're not doing the test. They're doing the test the new way, but not correctly because they didn't write it down. And so all documents need to be written. And this is critical for the accuracy and reproducibility of the laboratory tests, because that way everyone is doing the test the same way every single time. So in other words, do what you wrote and write what you are doing. So what makes a good document? So documents should be clear and concise. So try not to put too much extraneous detail that isn't really necessary for the test performance or the description of the process. They should be written in a user-friendly style. Remember, this is what staff are going to access to know how to do their work. And so it should be done in a way that everybody in the lab can understand and perform the test the same way. They must be explicit and accurate and reflect all of the step-by-step instructions and implemented measures, who's responsible, and how it's going to be managed. And they should be up to date. So remember, documents are living documents, and so they are changed. And when things change, they need to be updated to reflect those changes. And documents must be accessible. So in order for laboratory staff and pathologists to do their job the same way every time, they need to be able to reference all of the documents. So they have to be accessible. So any instructions on, and these are just examples, but any instructions on specimen handling, SOPs, any sort of quality control charts or troubleshooting instructions, safety manuals and precautions, all of these things need to be easily accessible to the staff, and the staff need to know how to access them. So the quality manual. So the quality manual, remember, is a component of the policies, and it's a document that describes the quality management system of an organization. So the purpose of a quality manual is to communicate information. It serves as a framework or a roadmap for meeting quality management system requirements. So all of those 10 quality management essentials, policies around those 12 essentials should be included in the quality manual. And the quality manual is really a reflection of the management's commitment to quality. ISO 15189 requires that laboratories have a quality manual, but they don't really, they're not very specific about how the style and structure, or how they need to be styled and structured. So laboratories should really construct a manual that works for them and works for their clients, their customers. So in terms of writing a quality manual, so when you're planning on how to actually put this together, it's really great to have a steering committee. So the quality manual really is the foundation of the laboratory's quality management system. And so because it really, you need to have buy-in from everybody in the lab, there should be representatives present at the writing process. So, you know, staff, clinicians, anyone who sort of interfaces with the lab and has a stake in the quality of the lab can be invited to be on this steering committee. You should state the policies for each of the 12 essentials of the quality system and describe how all of the quality processes occur, and make note of all versions of procedures. So the quality manual doesn't need to include all of the SOPs. That would be way too much, but it should outline the system for how the SOPs are organized, so that when you look at the quality manual, you should be able to find the SOPs and know where they are located. So the takeaways are there should be only one official version. It's never finished. It always is being improved, like all documents. It's read, understood, and accepted by everyone in the laboratory. It's written in clear, easily to understand language, and it's dated and signed by the management. And if you're looking for examples of quality manuals, the WHO LQMS toolkit has a link to a quality manual template that you could, if you're trying to write a quality manual for your lab, you can use that template to build off of. So next in documents are standard operating procedures, or SOPs. So these are written step-by-step instructions describing how to perform a test. So the purpose of SOPs is to ensure the following: consistency. So everyone performs the test exactly the same way every single time. Accuracy. So laboratory staff produce accurate results because they won't forget steps in the process. So if everything's written down, then no one will forget, or no one will be unaware. There's a way that they can reference what they're supposed to do. And so it'll ensure accuracy of the testing. And quality. So as a laboratory, it's our responsibility to produce consistent, reliable, reproducible, and accurate results. That's really considered the definition of quality, and SOPs allow us to do that. So what makes a good SOP? So they should be detailed, but also clear and concise, and provide directions for testing techniques. They should be easily understood by new personnel. So if once you hire someone into the lab, you should be able to give them the SOP, and they should be able to understand what they're supposed to do. They need to be reviewed and approved by management and updated on a regular basis. So here's just an example of an SOP format. It's by no means the only example, but this is an example of one that's computerized. And the nice thing about using a computerized system or electronic system for managing your SOPs is that you can store them and update them very easily. And so one thing that's really important about SOPs is that you use a standard format. So while the content may be different for different procedures, the format is exactly the same. So it should have a header. So I'm just going to advance all these. So you should have a header at the top that outlines sort of the name of the test, who the author of the test is, who the audience is, who validated it, the recipients. So this is meant for all of the technical staff. The version. So this is the initial version, when it was updated, and then where it can be found. Or sorry, so this is the version when it's updated and where it can be found. And then, so this is the header part, and then the sort of meat of the SOP will usually have a principle. So sort of explaining what the clinical background or methodological background of the testing is, a procedure for how to do the test. So this is the step-by-step instructions, and then the interpretation of the results. So there's a couple different types of standardized headers, complete and reduced. So complete standardized headers appear at the top of just the first page. And so here you can see these give the name of the procedure. This is the code that they use, or the numbering system, so that it's easily accessible, how many pages there are, that it was approved, what division, and then the date, the revised date, and then how often it is reviewed, or when the next review date is. I apologize. And then each subsequent page of the procedure will have what's called a reduced standardized header. So these just have, again, the name, the code for the header, the page number, and just the title of the header and the division. So this is just an example of how to use headers when drafting your SOPs. So now that you're ready so now I've given you an overview of an SOP, how do you actually go about preparing one? So it's really important to assess the scientific validity. So do your research ahead of time, make sure that the test that you're doing has been, do some research on the methodology of the test that you're doing, the clinical utility of the test, how it is used by clinicians to inform patient care. And then once you've done your research, drafting your SOP should involve including every step. So, for preparing SOPs, it's really critical to involve the staff who will be doing the test, because they can really explain to you all the steps that are involved. And it's really important that you include each one. You don't want to assume that any steps will be done if you don't write them down. Gather all your documents. So you have an SOP for how to prepare a peripheral blood smear. You might also in that SOP reference another SOP for how to obtain the specimen or how to release the results. So you want to be able to gather all the relevant documents to that SOP so that you can reference them easily. Establish a means for updating. So as we've talked about, documents, including SOPs, are living. They need to be updated. And so it's really important to decide how often you want to update them. So are we going to update them every 6 months? Are we going to review them for any updates every 6 months or every year? So that should be done at the outset. And then determine the procedure to use. So how are you going to store these SOPs so that folks who are in the laboratory can access them easily? How are you going to disseminate and communicate that information, et cetera? So what are the components of an SOP? So it should have the title or the name of the test, the purpose, so that's the medical use of the test, why you're doing it, instructions, and the instructions should really include the pre-analytic, the analytic, and the post-analytic. So pre-analytic include any sample collection or handling that's relevant to that SOP. The examination or analytic component are the step-by-step instructions on how to perform the test, as well as how to perform any QA along with the test. And the post-examination or post-analytic is how the test is going to be reported, what units are going to be used, what your reference ranges are, and decisions about what are critical values, if any. SOP should also include any references to verify that the method is established. So any of that background research you do to inform your SOP, you can include those references in the SOP. Must have the author's name and also the approval signature. So in our laboratory, we have the lab manager will approve and also the lab director. And it should have the date of when that was approved. So SOPs must be easy to read. So you have to remember what these are for, right? So these are for laboratory staff to access when they need to know how to perform a test. And so if you're in the lab, you have your gloves on, you're sitting there with the sample, and you're just need a quick refresher on how you need to do that test. You don't want to wade through a lot of words about the background and sort of a relevant detail to what the task at hand. So you really want to keep things concise, but include an adequate amount of detail. So I would say, like I said, I'm a hematopathologist and we have a flow cytometry lab where I work. And so if we were going to do a SOP on how to titer antibodies for our flow cytometry assays, we wouldn't want to include a bunch of background about what flow cytometry is, how the cell, like the methodology of the test in the purpose. The purpose should really be, this is the reason we're doing this test, to titer and optimize new antibodies that are received and verify the titer performance against currently used antibodies in the laboratory. So remember, just include the detail that's needed to perform the test. And a bit about manufacturer's instructions. So manufacturer's instructions are not a substitute for the SOP. So these can serve as guidelines and provide information, but they don't often include specific enough information for the laboratory to carry out the test. So examples of this include, they may not include any safety requirements that need to be included in an SOP. They may include they may not include any materials for testing. So an example, like in the flow cytometry lab, the manufacturer's instructions may say, incubate for 60 minutes in a dark area, whereas our SOP may say, incubate in a foil lined 15 milliliter conical tube. So they may not include all the information that you needed specifically about the materials if they weren't included in the test kit. And they're not going to often include any information about external quality control requirements. And so information from inserts can be used to inform your SOPs, but you can't just put the manufacturer insert in a binder and call it an SOP. You really need to draft an SOP that's specific to your laboratory. So job aids. So job aids are shortened versions of SOPs. They're handwritten or printed, and I think one of the important distinctions between a job aid and an SOP is that the job aids are visible at the location testing site. So, at the location of the testing site. So, whereas an SOP might be stored in a binder in the manager's office, a job aid is just posted at the bench, and so can be quickly referenced if there are questions. And it's a useful tool to have to ensure that all testing steps are correctly performed. So these are some examples of job aids. So they can be handwritten. They can be these sort of pictograms that show you exactly kind of step-by-step instructions, but in a visual format. And the important distinction, like I said, is that they are posted at the testing site. So one thing to keep in mind about job aids is that they are not a substitute for SOPs. And if you update the SOP, you also need to make sure those updates are reflected in the job aid. All right, so now that we've talked about all of these documents, SOPs, job aids, references, How are we going to make sure that they're all controlled and organized? So document control is a system that provides methods for formatting and coding documents so they're easily managed and accessed, and processes for maintaining the document inventory. So what's the purpose of document control? So a document control system is really critical to make sure that only the most current version of documents are the ones that are being used. So we want to make sure there's not a bunch of old SOPs that haven't been updated floating around in the lab that folks are using. The staff should really only be using the current version of the SOP. We also need a document control system also make sure that documents are available and easy to use when needed, and that they are appropriately archived when they need to be replaced. So here are some examples of controlled documents. So essentially a controlled document is any document that is produced and or used by the laboratory. And so the document control system really needs to incorporate and account for all of those documents. So these include SOPs, any references like books or articles that are used to inform SOPs or policies and processes. And then documents of external origin, and these are equipment manuals, manufacturer inserts, any regulations or standards that the laboratory uses. Those are all known as documents of external origin, and all three of these need to be controlled by the document control system. So what are elements of a document control system? So there should be a numbering system or a way basically of coding all of the documents in the system so that they're easily accessed. So similar to a library uses a very standardized code so that when you're looking for a book, you know exactly where to find that book, which aisle, which shelf. And so a similar strategy needs to be used for documents. They also need to account for an approval, distribution, and revision process. So how are documents going to be approved? Who approves them? How are they distributed to laboratory staff? And then when will they be revised? A master log to track all documents that are in circulation, because remember, we don't want old documents out there that haven't been updated. They need to make sure that have a policy for how they are going to be accessed at the point of use, and a system for archiving documents when they are old. So we'll go through each of these one by one. So for a numbering system, basically that refers to a uniform numbering or coding system. Like I said, that can be used to know exactly where to find documents in the system. So all pages of the document should be numbered, and you can use these are just examples, but you should use whatever process works for you and the folks in your lab. But one way to do it would be to use a letter for the type of document and then an incremented number. So for books, you might use a letter B as the first part of the code, and then each subsequent book is numbered 1, 2, and 3. For official text, you could use T, and then essentially you reference the document by the code, the pages, and the location. So if you were writing an SOP and you wanted to use, so if you're writing an SOP and you wanted to reference a certain book, because that was what you used to inform your SOP, you could use this code B2 188 through 200 BS1. And so this would tell you exactly where that reference was located in the laboratory. And again, you should use what system works best for your lab. If you have an existing system that works well, there's no need to change it. These are just some examples that you can use to organize yourselves if you don't have one in place. So this is an example. This was just that header that I showed in the SOP section. And so this would be an example of how to code, or what a numbering system would look like and where it would appear on a document. So next in the document control system is approval, distribution, and revision. So after you've prepared a document, then you need to make sure that it's reviewed on a regular basis so that you can decide, oh, there was an update, actually new literature came out, and it turns out we need to be changing the way we do this test based on this new literature. And so, you may not think about doing that right at the time that you read that article. And so it's really important to have a set time where you review all of the documents and then you update them as needed. once you have the documents reviewed and you revise them, then they need to be approved by a manager and director. And then they need to be formally issued and distributed to staff. And so this is just an ongoing, the sort of review, revision, approval, distribution is a circular pattern. You should just continue to do this in the lab to always keep documents updated after you've prepared them. And your document control system should outline how that is done. So lastly, our master logs, accessibility, and archives. So a master log tracks all the control documents that are in circulation. So a way that you could do this is keeping a log in the binder. So if you have a binder of all the SOPs that you need to do hematology testing, then you can have a log at the beginning of that binder where folks can check out procedures. So if they need to check out the procedure for doing a ESR, for example, then you can have a log and say, okay, so Joe Smith checked out the ESR procedure and then when it was checked back in. That way you're keeping track of all the control documents that are in circulation so that there's no documents that are missing and there's no documents floating out there that you haven't accounted for, so that when you go back and do your revision or you update anything, there's not old things that out there circulating that you missed. So again, documents must be accessible because folks need to use them to do their jobs. So you need to figure out a way, and a document control system should outline how the staff can access any documents they need when they're performing their duties. And it's important to know that some of the duties related to the laboratory and laboratory testing are done outside of the lab. So any sort of phlebotomy that's done on the floor, for example, you may want to have a process or procedure for how you're going to manage any SOPs around phlebotomy that live on the floor or the wards. And then you need to have a system for archiving. So like I said, old documents must be archived. You don't want them floating out there, but you also don't want to just throw them away, because old documents can be really helpful for creating new documents or troubleshooting problems that you notice later, or reviewing your quality practice. So you want to have a system for archiving all of those old documents and define a period of time that you're going to keep them. All right, so implementation. So now you're ready to implement your document control system. What are the steps you need to take? So first, you need to collect all the existing documents and records. So anything floating out in the laboratory, any random SOPs, any manuals, all that stuff needs to be, any package inserts, all those things need to be collected and cataloged, and you need to figure out if you need to review, and you need to review all of them and figure out whether you need to update them or not. So you'd be surprised, I think, once you go through this process, how many outdated things are out there floating around in the lab. So it's really important to kind of go through this, catalog everything, and update them as needed. And then once you've done that, then you can see what your additional needs are. So you can see where the gaps are, like, oh, I didn't have an SOP to describe how we were going to disseminate this test result to the floor. So we need to draft an SOP for that. So that's another good reason to basically take an inventory of all the existing documents you have in your laboratory. You need to develop or obtain documents, forms, worksheets, logbooks, and reports. So, so part of this determining additional needs is filling those needs. So if you say, oh yeah, we needed a form for how we were going to track specimens through the chemistry lab. So now we need to generate that form. So then you could go and say to your colleagues in the other chemistry lab and be like, what form do you use? And so just kind of obtaining all the information you need to update all of those documents. An important part of implementing any sort of document control system is involving stakeholders. So the folks who are going to use the documents really need to be involved in this process, because they're the ones who are going to need to access it. They're the ones who are going to need to use them. And so it should be easy for them to use and clear, because otherwise they won't use it. And we'll have a situation where people are doing different things and not following the step-by-step procedures. So what are some common problems in document control? So laboratories that don't have a document control system or have a poorly managed system have, they may have outdated documents in circulation. So like I said, if you didn't do that step where you collected all the documents and reviewed them all, you may have some SOPs floating out there that are reflect the old way of doing things, and but folks might still be using it because they didn't know that the documents were updated. You might have multiple copies in circulation, so that becomes a problem when you need to update them. So kind of corralling all those copies, updating them all, and then recirculating them can be challenging. And so trying to keep a system where you have only the number of copies you need in circulation is important. And failure to control copies of external origin. So these are, again, those manuals, the package inserts, the regulations. So if you don't have a way of sort of cataloging and controlling those, they can also get lost or just be filed in a random drawer somewhere, or maybe you have someone at the bench who's using the package insert instead of the SOP. So you need to make sure you're not only controlling the documents you created, but the documents that you've accepted into your laboratory that you use to inform your SOPs. So that's all for documents. So why don't I pause there and see, before I go into records, and see if anyone has any questions.
Yes, Emily, could Dr. Glynn, I should say. There, we have a question here.
Sure.
This one, though, is from earlier in the lecture. It says it's by Mr. Sinji, who asks, in the case of reagent inserts, how is it possible to have them controlled, especially if the lab changes reagent type frequently and a variety of reagents, question mark.
Yeah, so I think it's just I guess how to best answer this question. So I think the key is just that you have a way basically of reviewing all of these reagent inserts. So once you have taken the reagent in the lab, you've done your checkout and validation. So let's say you get a new reagent, you've done your checkout and validation. You want to have a way basically of filing that reagent insert away and to document that you have incorporated the reagent insert in your SOP. So let's say you've gotten a new reagent for your hematology analyzer. You basically want to check out, document you've done that validation, record that validation, file the validation paperwork, and then also update your SOP. So, because you've changed your reagent, the SOP might change. And so you want to reflect that in the SOP and then reference in the SOP that this is reference the new reagent insert. And so I guess that would be a way that you can account for changes in reagents. Do you have any other thoughts, Tim?
No, I think it's a really hard question. And I think this is why bringing these concepts to the reality on the ground is important. I think that's what you suggested is the only thing I can think of. It's tough.
Yeah, I think it's one of those things that the document control, I think, takes a lot of upfront work. But I feel like once you've kind of done it and you get in a rhythm of doing it, it gets easier. And also you have a system in place to sort of just a routine for how you're going to do these things, and just keep track of everything. And so then I think it does save time later, because once you're like, let's say you have staff who's working with a new reagent and they're like, wait, hold on, this is new. Like this procedure I can't do this procedure because the reagents are different. I'm not sure what to do. Then, they spend a lot of time trying to figure out what they're supposed to be doing because nothing was updated. So I think it does take some time up front, but it does save some time later on.
Yes. So, okay, we got two more questions, but okay, one of them, I'll ask those two and then I think we'll move on. The second one was by an anonymous attendee, and it says, why not lump documents and records together under document control?
Yeah, so great question. So I think, so there's some specific differences between documents and records that make them, that make it so that you kind of store them differently. So I think one of the things that is different, so I'll just give an example, and we'll talk a little bit more about this when we go over the records, but documents are living, right? So you need to update them all the time. So you need to have a procedure or some sort of outline for how you're going to keep them updated, review them and keep them updated and disseminate the updated documents. Records are never updated, right? So those are just a reflection of what happened in the laboratory. So you don't need to have a procedure for how those are updated. You just need to have a procedure for how they're stored, how long you're going to store them, et cetera. So there are some differences in documents and records that require them to be stored a little bit differently. I think that there are some, I would say like a document control system should, can incorporate information about how you store records, but there's just some nuances about how they are handled that make it so that they're different. They need to be handled a little bit differently, if that makes sense.
Okay, that's great. Yeah. I hadn't really realized that, how different those two are, but that makes sense. This question is by Mr. Nyaji, and he asks, how do we maintain the integrity of QMS documents when changes are implemented?
Great. Oh, sorry.
No. I was just reading it to myself, but yes.
Okay. Great question. So, so I think that's where that's why the document control system is so important. And I realize, depending on the resources of your lab, it can be really challenging to maintain the integrity of the document. So I'll give you an example. So our lab uses an external document management system. So everything is cataloged. It's all computerized. So if someone changes the document, it automatically timestamps when the document was changed, and it requires that it be signed and documents the signature and time when it was signed. And then it also only allows one person to check it out at a time. So I recognize that can be really difficult for all labs. This is a really, it's an expensive software. And so it's not totally applicable, but I think you can sort of take aspects of that and adapt them to your resource level. So, if you have a quality manager who, it's their job to manage all of the quality documents, they can be in charge of the document control system and organizing it, making sure that all the binders are up to date, having the log, like I mentioned. So having the log of when things are checked in and out is really important. And then also, one of the ways that you can really maintain the integrity is just documenting. That sounds redundant, but all of your SOPs, procedures, they should have a section where you say, this is when the original date that it was drafted. This is when it was revised. This is the next revised date. And so that's another way that you can maintain the integrity. And also, always making sure that documents are signed off on and dated. And so if you create templates for SOPs that already include that signature, who needs to sign it and who dates it, that's what we do. So we have a standard template for all our SOPs. And at the bottom, it has a line where the director signs it and the manager signs it, and it has the date that they signed it. And so you won't forget. So you'll always remember, I need to sign here. And so that's another way to sort of maintain the integrity. And so that way, if it's not signed, how do you know it's reviewed? But if it's signed, you know that it's reviewed. So I think that there's some things you can do, depending on what your resources are, to maintain the integrity by sort of templating things, making sure dates, times are documented, and also making sure to keep track of the documents.
Okay, I think that's a good spot to go back to the lecture. I did want to say, Ken, there are some questions that we'll need to answer to the whole group at the end about certificates and also some of the lectures that were missed earlier in the session. But Emily, I'll mute now and turn it back over to you.
Okay, sounds great. All right. So now we'll talk about records. So records are permanent information produced by the laboratory when performing a test. So why are records important? So they provide continuous monitoring. So they allow the lab to know, what's going on, how are we monitoring our performance? So things like turnaround times, Levy-Jennings plots, that's another way you can see, over time how your lab is performing. They track samples. So they allow us to say, if something went wrong, they allow us to go back and say, oh, okay, now we need to troubleshoot. We need to do our root cause analysis. We can do that because we've tracked a sample and kept adequate records. They're important for evaluating problems. So if you have sort of a systemic problem in the laboratory, you know that because you've kept really good records and that problem keeps coming up over and over again. So you can kind of address that. You have the data to evaluate problems on a more systemic level. And they're a useful management tool, having access to turnaround times, having access to, how many, when the QA is performed, how well you're doing on your proficiency testing. Those are all things that are important for managers to know when running a laboratory. So I like to think of it as documents really tell us why and how. So they tell us how to perform the testing. Records tell us what was actually done. So records are really what the laboratory did. It's sort of, so I basically like to think of it that way. Documents tell us why, how, records tell us what was done. So these are all examples of records that I've encountered. So this is a logbook. That's an example of a record. This is a proficiency testing result. This is LIS. So this is an example of records that are going to just be disseminated directly to clinicians. This is a report, a flow cytometry report. So this is a record. This is a laboratory worksheet or for sample tracking. This is an example of records. So these are all encompassed under records. And as you can see, there's a variety of ways to maintain and keep records. So they can be handwritten, they can be generated by a computer, but sort of rendered as a report, or they can be directly computerized and disseminated in a computerized way. So here are just some examples of records. So as we talked about, logbooks or registers, workbooks or worksheets that are used in the laboratory, instrument printouts and maintenance records, all those, when you record the temperature of all the refrigerators and freezers, that's another, and water baths, that's another example of a record. Any quality control data that you collect, proficiency testing, patient test reports, personnel records. So anything in the sort of human resources department, that's an example of a record. Internal and external audits. So if you were visited by an accreditation body, their summative report would be an example of a record. Any sort of continuous improvement projects that are done by the lab, incident reports, user surveys, and customer feedback. So that's one we don't often think about, but if you survey your clinicians or get any feedback from patients, that would be another example of a record, and any sort of critical communications. So again, these are all basically different manifestations of things that were performed in the laboratory related to lab testing. So here's an example of a lab worksheet. So this is actually a worksheet that we use in our laboratory. This was the old version here on the left, and this is the newer version here on the right. And so essentially, this basically allows us to say, the old version was okay, but it wasn't great. So you would just basically circle this is for flow cytometry, you'd circle the panel that was done, and then staff would initial based on who accessioned it, who processed it, who set it up, et cetera. So then we figured out there were some gaps. So we tried to do some root cause analysis of different problems that happened in the laboratory. And we realized, this record or this template is just really incomplete. We can't always troubleshoot, or it's not easy to troubleshoot issues based on how this is set up. So one of our leads revamped the sheet, and now it's a lot better. So essentially, so you don't just choose the panel, but whoever runs it needs to initial here, who accessioned it, who processed it. So all these require initials. And then obviously it has all the pertinent information about the test, the patient's demographic, what type of sample, et cetera. And so this is an example of how we use it. So this was an example of a tissue that was run for B and T cell flow cytometry. And so it tells us basically that, so it allows us to investigate. So if there was a sample switch, then I can say, oh, I need to ask Brian, that's BM. So I need to ask Brian because he's the one who accessioned it. Or I need to ask Gina, who's the one who processed it, what happened if there was a sample switch. If it looks like I look at the data and it looks like maybe the wrong cocktail was added, then I could go back to Gina and say, Gina, it looks like you set this up. You ran the B and T cell tube. It looks like the wrong cocktail was added. can you help me troubleshoot? If it looks like the wrong data was inserted into the template, then I can look at who analyzed, so who actually uploaded the data into the software. And that was Sue, so that's SG. So I can go to her and be like, it looks like the wrong template was used. What do you think about that? And then if it looks like multiple, we go throughout the day and we're like, multiple cases look very strange. There's some compensation issue. Then we know, oh, we can look at what instrument it was run on. So we have a space for that. And it basically, we can say, oh, there's something wrong with instrument A, we need to adjust the lasers. So this really allows us to go back when we identify that there are problems, go back to who the person in the lab was who might know more about it and help us troubleshoot, and also what instruments were done. And it helps us say, it's also a check. So it's also a good checklist for the staff to say, okay, have I done this? Have I verified the patient identifiers? Did I check the paperwork? Did I do the charges? And so I would encourage you, especially if you do, testing where multiple people are sort of involved in the process or procedure, to use one of these worksheets to kind of help you record what was actually done and who did it. So these are records that are often forgotten about. So disposition of rejected samples, referrals of samples to another laboratory. So let's say you send a sample out for TB testing, because you don't do that testing in-house. You need to keep track that you sent that sample and when you sent it. And so you can follow up on the results. Records of adverse occurrences or problems, inventory or storage records. That's a pretty common one to forget, but very critical for tracking, supply issues. Equipment purchase data, preventative maintenance, and troubleshooting. So all this to say that there are a lot of records out there in the laboratory that we don't, it's kind of hard to think like, oh yeah, that's a record. And so when you're creating your control system, so your document and record control system, I suppose, it's really important to establish a process for what you want to record, what information and data you want to collect, and a way to collect that data, because that way it'll help you not, avoid forgetting about some of these ones that are easy to forget, that we don't necessarily counter every day. So this is just a summary of what's required on test reports by ISO 15189. So we'll just quickly go through. So things like test ID, what laboratory it's performed at, any sort of patient, obviously the patient unique identifiers, who requested the test, the date and time of collection, the time it was received in the lab, the date and time it was resulted, the sample type, any sort of reference ranges, and any interpretive comments. And if you amended the report or corrected it, you also need to note that you corrected the report, the date, and we always say what the original result that went out was, and then what the corrected result was. So on to document, record, and storage. So the key components of document and record storage are permanence, accessibility, security, and traceability. So there's a few ways to store documents. So you can store them on a paper system. And so the way that these four aspects would look like on a paper system is permanence basically means that you don't just have a bunch of loose papers floating around the lab. So they need to be in a bounded book or in a binder. The pages should all be numbered and coded according to your document control system. And you need to use permanent ink, no pencil, no erasable pen. They have to be accessible. So, if you're using a paper system, that can be a little bit difficult, the accessibility. So just making sure, again, everyone in the lab is clear on where the binder is stored, having a logbook so they can check out the procedures, those types of things. Security, so the documents need to be stored in a secure location, and particularly records, since those have patient information, need to be maintained in a place to ensure confidentiality. And of course you can store, documents and records might be stored in different places, but for the records in particular, that's very important. And you also want to keep it safe from hazards. You don't want to keep your only copy of your QA data log on the bench next to the bleach in case the bleach falls, and then all of your QA data for the past 3 years is lost because the bleach fell on it. So you just need to make sure that you keep it away from hazards in a safe place. And then traceability is really important. So I think that kind of goes back to that worksheet I just explained. You want to make sure that you create a record system that allows you to collect the adequate data, so that if there's a problem, you can go back and easily trace it throughout all the processes in the lab and figure out what went wrong. So if you're using an electronic system, some of this is a little bit easier. So permanence, so because the electronic system is accessible on multiple computers, you just need to have a way to back it up. And have a regular, I think the important part of an electronic system is that you have a dedicated IT person or someone who has some knowledge of IT to maintain the system. Accessibility, so typically computerized methods are a little bit more easily accessible if you can put a computer at each bench. But on the flip side of that is that you have a lot of people kind of in and out accessing that information. And so you really need to make sure that things are password protected, that the workstations are password protected, that people log out when they're done using it, et cetera. And traceability. So, I think this is one situation because you don't have a paper trail where electronic systems can be a little more difficult. So you want to be able to use a system that allows you to trace samples or trace changes that were made and doesn't just kind of override them without acknowledging how they were changed. So retention time, this is the last thing, and this really applies to records specifically. So, because we talked a little bit about in the document control system, archiving and things like that. But for records, you also need to decide how long you want to keep records, and this will vary by lab. So remember, the purpose of records is to have documentation of what was done at the time of testing. And so the length of time that you keep the records may vary based on how long you need to access them. So you say, okay, we're gonna review a year's worth of Levy-Jennings plot. Then you want to keep your Levy-Jennings plot for a year. So just how long you decide kind of how long you want to, how long ago you want to do your retrospective review, and then decide your retention time accordingly. A retention time can also be dictated by government requirements or standard requirements. So accreditation bodies typically have set time that you need to keep records. Research, so if your laboratory is involved in any sort of research, that may also dictate how long you keep records. And then time between assessments and audits. So if you are getting, your lab gets inspected every 2 years, that also is going to dictate how long you keep some records. So just to summarize, documents are written policies, processes, and procedures that tell us sort of why we're doing things, what we're doing, how we need to do them. Really, how the testing is done and why we're doing it. These are living documents that must be updated and maintained. In contrast, records are information captured during the process. So this is telling us what was done in the laboratory. These are permanent. They do not require updating and really should not be updated. So you don't change a record. If you want to make a note of, that you troubleshooted something or that you went back and you looked at it, then you can sign and date or make a note on the record of what you did and then sign and date, but you shouldn't ever change the record. And document control systems basically are critical to making sure that the most current document version is in circulation and that documents are available and easy to access by those who need them. So just to close, information is what we produce, right? So as a laboratory, we produce laboratory information that informs very important patient care decisions. And so it's really important that our the purpose of all of the lectures I hope you've taken away is that the purpose of laboratory is for laboratory testing to be reproducible and accurate and done the exact same way every time. And so in order to do that, you really need to have a strong document control system and have a way of managing documents and records in order to make sure that that is done. So that's all I have for the lecture. Before we move on to the case, were there any more questions?
Yes, there were 3 questions and now there are 4. The first question is sort of a general one. I'll just answer it. So the question, he says, this is by an anonymous asker. Is it necessary / compulsory for every staff to go through the quality manual and all SOPs? Yeah, I would say yes to the quality manual, and they should go through every SOP that applies to the work that they do. Emily, do you have any other?
No, yeah, I totally agree. I think, the quality manual is really the mission of the lab. And so they need to review the quality manual. They don't need to review it every day, but they should review it periodically and certainly every time it's updated.
Yes. So there's a second question from that same person. So let me ask the others first. So the next question here I'll go to is by Ms. Tanko. And she says, is there any duration on how often SOPs/quality manuals need to be updated?
So I would say that you should decide for your laboratory the appropriate interval. I don't know, Tim, I don't know, do you know, I'm sure that some accreditation bodies may have some guidelines.
I haven't on there is a possibility that ISO has guidelines, but I haven't, the only guidelines I'm aware of are CLSI, and even those are not considered rules, they're considered guidelines, and it's for review every year for the quality manual, and at least every other year for the SOPs. In most labs I've been in, we review both every year. So you don't necessarily have to update, but you need the medical director and or the administrative director will review and sign and just say, yes, this SOP still matches what we're doing, our practice, and is still, updated, or they'll update it during that process. So I would say every year, at least.
Great, thank you.
The third question here is from Ms. Sanyanji again. It says, is it acceptable to amend records? And if yes, what is the best way to amend records? And then he elaborates. He says, example, we conducted management review and documented minutes and distributed to participants a month later. A month later, I discovered I forgot to include one of the discussion in the minutes. Can I amend the minutes and include the missing discussion and redistribute? Question mark.
Yeah, that's a great question. So yeah, so like we said, so records typically are not changed. I will say, I think, so for that situation where it's sort of an update to the minutes where where something was left off, I think you could just sort of addend it, kind of add it at the end and say this, this was left off, and then note, the original time that you sent the minutes and then when you addended it. I think for things like patient records, things like that, when we do what's called if there's an error, we'll do what's called an amendment, because we don't want to necessarily to addend something that's to an incorrect report. So if we're actually making a correction, we'll do what's called an amendment. So we will issue a new report with the correct result, but we will say in the body of the text, this was amended because of this, and we'll quote the original result. So we always quote the original result. So I would say, it kind of depends on what record you're talking about. I think if you're talking about things that can just be added on, or notes that you want to take in response to a record, you can just sort of note that and then sign an initial and date it. If it's something that's like a patient care issue, you may need to actually issue a new report. I would say you're not going to change the existing record. You're issuing a new report and then just saying what the old report said in the new report. That's how I would handle that.
Okay, that's great. So then the last question says, in laboratories that only do electronic record keeping, can the lab keep only ESOPs? Yeah,
so that's what we do. So all of our controlled SOPs are kept in a software that basically only allows one person to check them in and out. You can view them so you can't change them, but you can view them. So if I'm like, oh, I have a question about, how to do B and T cell clonality, then I can just look it up in that, in the software and look at the SOP there. I think if you do an electronic SOP system and there's not a computer at every bench, so it's going to be difficult for staff to access computers, I would say that's when you might want to use a job aid. So you might want to print a job aid that sort of has an abbreviated SOP on it and post that at the bench. That way, folks don't need to stop everything they're doing, go to the computer, log in, look up the SOP, figure out what they need to do. So I think that's where job aids can be really helpful. Just the thing you need to remember about job aids is that because they're posted at the bench, they're not kind of where all the main documents are kept, that they need to be, when there's an update to the SOP, you just need to make sure to update the job aid as well.
All right, those are all the questions for now. You can continue.
Okay, great. All right, so I'll move on to the case. So this was sent out to you earlier. And as Dan mentioned at the beginning, if you have any thoughts about the case or you want to record your answers so that I can discuss them live, you can put those in the chat, not the Q&A. All right, so you are a newly hired manager for a laboratory that participates in T-cell subset proficiency testing. On one of your recent surveys, the absolute NK result was out of the limits of acceptability. Your director notes that response that this is unacceptable and on the results form and files it in your EQA binder. So this is a copy of your EQA report. This was the unacceptable result here. And then this is what your director has sort of the response to the unacceptable result and what's going to be on kind of kept on record about this, your laboratory's response to the result. So EQA and PT results are an example of a document, a record, or none of the above. So give just a few seconds if anyone wants to put their answers in the chat. All right. Okay, so Tim, were there any?
Sorry. Yes,
Oh,
they
sorry.
just started pouring in all of a sudden. We have eight or 9 people and they all say record. So
records, perfect, exactly,
yeah.
Great job. So this is an example of a record. So EQA, PT results, right? This is what the lab did when they performed the test. So that is an example of a record. All right, so after we've done this, you're flipping through your EQA binder and you notice this response. And you have some concerns. So what aspects of this record are problematic? So I'll just give folks a few seconds to respond in the chat. And then Tim, whenever you're ready, or.
Yeah, I'll let you know. It's still cooking. I think it takes a while to come through. What aspects of this record are problematic? Okay. So we haven't had, nothing has come through yet. I'll give it 10 more seconds. And
okay.
Then,
yeah, and then we can just discuss.
Yeah. Okay, nothing has come through yet. Can I
Okay.
try?
Oh, sorry. Yeah, go ahead. Yeah, I'm sorry. Go ahead. Go ahead.
You already showed the answer.
No, go ahead. Go ahead.
I was gonna say there's no dates, and it's basically inalleged. I was gonna say the main thing is there's no dates. It's not clear what they're saying, or who signed it. So I just don't know. Yeah. Oh, someone wrote that not clear, too much text, illegible handwriting. Yeah, so I think it just takes a while for the answers to come through. So we just have to wait longer. But people wrote in not clear, too much text. These are different individuals writing. Illegible handwriting, no signature or dates.
Perfect. Yeah, exactly. Yeah, so it's illegible. I don't know what they're trying to say. So it's important for records and documents that they be clearly communicated, right? That's critical. And for records in particular, especially if you're in a place where you're doing things by hand, so you have a logbook that's by hand, in this case, you're responding to your EQA results by hand. I would try and enlist someone who has legible handwriting to write the things down and keep track, because it's there's no way. I mean, if you can't read it, then you're sort of starting from scratch. It's like it didn't exist. And so so I'd say, everything needs to be legible. And this is especially important if you are doing things by hand. Again, there's no initials and no date. So that's right. So any change to a record, any updates to a record need to be initialed and dated, so we know who made that change. And it's incomplete. It doesn't really allow, like there's no information here about how the testing was done, what sort of steps were taken to evaluate the problem. It's very incomplete. So if you're going to, so I think one aspect of records that's really important is that it be complete, it be a reflection of what you did. And so if you can't tell from the record what exactly was done, then it's an incomplete record. All right. So you were lab inspected by CAP the next year, and they asked to see documentation of the unacceptable PT result. So what's the best response to the inspector's feedback to the all common checklist 1600? So I'll just read it to you, although their handwriting is a little bit better. So their feedback for the all common checklist item 1600 was PT integration routine workload. The laboratory was running PT in duplicate once on each instrument. However, patient samples aren't routinely analyzed in this manner. So how would you respond to that feedback? What steps would you take in your laboratory?
Emily, do you mind reading aloud the 4 options?
Yes,
here,
sure.
A, B, C, D?
Yeah, so option A is to provide laboratory staff with reference materials on NK cell reference ranges. So basically just educate them on what NK cell reference ranges are. B is update the existing SOPs to provide clear and concise instructions on how to run PT material. C, create a job aid of the existing SOP and make this available at the T-cell subset bench. Or D, update the SOP with instructions on how to run PT, but keep it safely locked in the manager's office so staff can't access it and make changes without the manager's permission.
Okay, so we have, answers have started coming in, and the first person says B, second person says B. So let's see if there's any more. So we have 2 votes for B, 4 votes for B, none for any of the other. Okay, 2 more seconds and then we'll. Okay, someone says B or C, which is kind of my thinking. 2 more Bs, so we have 6 or 7 Bs and 1 B or C. Another B coming in. So the overwhelming majority is saying B, and then another B and C.
B and C. Oh, okay.
Yeah. So 10,
yes.
I would say 10 Bs and then 2 B and Cs.
Oh, okay. Okay.
Yeah.
So B was the answer I was hoping for.
Yeah.
So update basically. So as this isn't a case about PT, but one of the principles of proficiency testing is that you run it exactly the same as patient samples, and that's not what was being done in this particular laboratory. And so so that was the feedback that we got from the inspector. So what needs to be done? So you need to update your SOP in response to this feedback to provide instructions on how to run PT material. So the prior SOP said to run it in duplicate on your 2 machines. The current SOP needs to say you run it exactly like a patient sample. So that's what needs to be done. I think the providing laboratory staff, educating them, that's always a good answer. I think that's it's always good to provide more education than less, but I'm not sure just providing that education or providing them with reference materials would address this concern. C, so create a job aid. So I think that's really good. I think it's always good to create job aids. I just wouldn't create a job aid of the existing SOP because it was incorrect. So that was So the important thing about job aids is they need to reflect what's in the SOP. So once you've updated the SOP to run it like a patient sample, then you could create a job aid to reflect the new SOP. And so D is the other one I think was, not necessarily wrong, but the reason I didn't like D is because SOPs need to be accessible to folks who need to use them. And so if it's locked in an office somewhere and the manager has to stop what they're doing to unlock the office so you can access the SOP, or, what if they're on vacation, then, people are stuck. And so I think especially if you're keeping paper documents, or paper copies of SOPs, that's how you're managing your documents and records. I think it's important to keep in mind where you're keeping those documents. So of course they need to be, in a secure place, but locking them away in someone's office is maybe not the best idea because then people can't access them. All right. So in response to the inspector's feedback, you as a quality manager, create an unacceptable proficiency results investigation form. So in what ways is this form a better record than the original response to your lab's unacceptable PT result?
Okay, so you want them to just type free text
Yeah,
to
yeah,
the chat.
yeah.
Okay, okay.
Yeah, we can just give a few minutes. If people don't have any responses, that's okay. We could
No,
just discuss some of the responses.
we have an engaged crowd here. I'm sure that.
Or if you have questions, you can ask questions.
So, okay, the first just came in and he says it is legible and permanent. The second one says legible. Signed. Someone else, clear, explicit signatures and dates management review. Next person says legible, place for dates and identification of reader and checker.
Perfect. All right.
Yeah. Someone more coming in. Clear, can be tracked, legible and signed. Legible. These are just different people writing in.
Okay. Yeah. So I totally agree. So I think it is better because it's legible, it's organized, it's easy to understand. There's, as people said, there's space for defined initials and dates. There's space defined for initials and dates. So, so like I said, I think this is one of the ways. So this is actually a template that we use in our lab, and the template, I think that's one important aspect of creating templates, because then it's easy to remember what you need to do. So here we've created, oh, go ahead.
Sorry, Emily, sorry, it's the last two that just came in, I had added more to what,
oh, okay,
which I liked. So is it, one person, I think the first name is Bake, says detailed information about the problem. So I like that. And then
yes,
someone else, I can't see the name too well, I feel like it's Ifoma, but I'm not sure, says shows step-by-step measures to address the query. So I like those two.
yeah, that's perfect, exactly. Yeah. So this scenario, sorry, a little hard to go back. So this scenario really has no information about the investigation, and if it did, we wouldn't be able to read it anyway. But yeah, I doubt it does, because it's just like 2 lines. So this doesn't really tell us what was done, what went wrong, how we investigated it, what we did to address the unacceptable results. Whereas this one really outlines, as the participants mentioned, really outlines what we did. So we you know, it was reviewed, we looked at the gating, we decided that was acceptable, we reviewed other data collected, we reviewed the QC data for that day, we reviewed prior and subsequent PT data. So everything, you know, all the steps we went through to see what was done, we did. And it also says sort of what became of that evaluation, what our interpretation of it was. And like we mentioned before, it's legible, it's organized, and it has a space for someone to sign and date. So this is an example of how templating can be used to really guide sort of next steps. So the template, or the standardized form is how we call it, is an example of the document. And then once you've used the standardized form and filled it out, then it's a record. And so standardized forms can be really helpful to sort of guide what steps you need to go through, and make sure that you have all the proper documentation. So I would really encourage you, if you're sort of coming up with how to manage your documents and keep better records, one strategy you can use is the standardized forms. All right. Any other questions? All right. Well, thank you all so much for attending this 10-week course. We really appreciate it. We hope you found it helpful. And please fill out the feedback survey so that we know how to make it better for the next time. And the questions at the end. You know, we really enjoyed having you. Thank you all for being so engaged and for participating. I know it's hard in these virtual formats to participate, but I've been really impressed with the great questions people have asked and the comments folks have made. So really appreciate your engagement throughout the course.
Absolutely. Thank you, Emily. Thank you.
Don't log off yet. Okay, thank you, Ken. I was hoping you'd come on.
Yeah,
So there were two questions that
okay.
to draw your attention to before. I don't know what else you're going to announce, but on the top of the chat, there was a question about the Tuesday session and also uploading the session on organization and case review. And then there's a question about certificates also.
Yes, I was just about to get to that. So
Okay.
on behalf of ASCP, I do want to thank all the participants and of course all of our faculty as well. We have be looking in your email because we will be sending out course evaluations and also a post test. And following that, the last two sessions, the one from Tuesday and the one from today, will be uploaded on the landing page. So if you missed either of those, you can view those there before you take the post test. And we will be emailing certificates probably the second week of June. But be checking your email for those follow-up pieces and action items. Timothy, Emily, do you have anything else to say, or should we wrap it up?
Just about the course, there's a concern that the Tuesday course is not open or something like that. Is that
it takes us a couple days to load those. So the one
Okay.
from this past Tuesday, it hasn't been uploaded on the site yet, but it should be
Okay.
up there today or tomorrow.
Okay.
And then today's should be uploaded by the end of the week as well.
Okay. All right. I just wanted to add my voice to some thanks are coming in the chat. This has been a great course. It worked out really well. So I thank you all for sticking with it. 10 weeks is a long time. And yeah, it just seemed like it was a big, kind of an ambitious course, and we weren't sure how it would work, but it seems to have worked very well. So thank you so much for attending. And please provide feedback so we know what to do next time. And yeah, and that's all. Just thank you so much. And thanks to all the contributors who gave the lectures and to you all who attended. Yes.
Thank you.
So,
Great.
yeah.
Thanks to all of you.
Thank you.
Bye-bye.
Bye.
Bye.